I'm going to be talking about the work that I've done over the past few years here at USC, and it has been on synchronization and carpets of cilia. So this is a brief outline of the talk. I'm going to basically go through each keyword that I mentioned in the title of my presentation: synchronization, ciliary carpet, simulations and theory. That's basically the outline of the talk. So I'm first going to start off with by motivating the problem. Why do we look for what's special about ciliary carpets, what's synchronization, and so on. So synchronization is basically a phenomenon that's there everywhere in biology. We notice it everywhere, we see it everywhere, we experience it in fact. And some of the most common examples are a bunch of fish schooling or birds flocking. So in all of these examples, synchrony involves agents with individual freedom tuning their behavior to sort of give rise to these collective behaviors or patterns. So fish and birds are examples where each entity has its own freedom, but the agents do not have to basically show synchrony in space alone. They don't have to move. For example, the video next to it in the third frame is a bunch of fireflies, which actually are sitting stationarily on trees and forests. And they coordinate the flashing in such a way that you see these wave-like patterns emerge. And something very similar happens even in the human brain. There are neurons and even the pacemaker cells in the heart, where if you put these individual guys in a petri dish and you actually measure the voltage, you would see periodic spikes. But you put 10,000 of them together, they somehow know to synchronize their beat. So these are some very popular examples. But the first scientific inquiry into studying synchronization was made by the Dutch scientist Christian Huygens, who basically had this setup here, which involved two pendula which were mounted on a wooden plank mounted across chairs. So this is a modern realization of the same experiment. I'm going to play this. So each of these are started independently in random order. And what we see is that slowly pairs of them begin to synchronize. The guys towards the end is a little slow in getting the message, but eventually that guy is going to get there as well. Right? I'm going to speed this up a little bit. So they start waiting in synchrony. Now, this is a beautiful example which showed that synchrony can emerge purely from physical mechanisms alone. There is no need for a leader like a human orchestra. There are many more such examples which I had fun reading in this book called Sync by Steven Strogat. I in fact took it while I was taking the nonlinear dynamics course with Professor Newton. So highly recommended exercise for people looking to get into this field. So all the examples that I showed so far were synchrony at a macro scale, but there are plenty of examples at synchrony at micro scale as well. And the one that we are particularly interested in for today concerns cilia and flagella. So cilia and flagella are these tiny hair-like organelles that protrude from cells. They're about 5 to 10 microns in length. And what they do is they have an intricate internal structure. They're powered by motor proteins, and they are able to execute this back and forth oscillatory motions, and they coordinate across scales, like in the scales of cells and tissues, in what is called this metachronal coordination, which I'll talk about in just a second. So from the individual level of cilium, you see collective behavior emerge in huge carpets of cilia. So why is this important? Well, we all know that the human body, although symmetric on the outside, actually is asymmetric inside. For example, most of us have our hearts on the left side, pancreas on the right, and so on. But scientists have found that there is, in an embryo, there is a little patch called the Hansen's node, which has tissues with cilia on them. And what these cilia do is they have this swirling kind of motion, and this helps them set up a directional flow. And what was found quite astonishingly is that when the scientists went in and actually stirred it in the opposite way, the mouse embryo, which is actually what is shown here, the baby mouse ended up with its body plan entirely flipped. So this is quite astonishing that this flow that cilia induce sort of leads to this left-right asymmetry in humans. Now, they're not only important at birth, they're also they're throughout the body in humans and help us execute a lot of day-to-day functions. Like cilia are there inside our respiratory tract, and what they do is they coordinate across, like there are carpets of cilia, a lot of them, and they beat in such a way as to, you can already see this here, this global wave-like motion, which pushes the mucus, which contains dirt particles. So this acts as the body's first line of defense to keep the respiratory system functioning and healthy. These are a couple of examples, but then cilia are also found in the brain, where they serve to push or pump cerebrospinal fluid. They're also there in the male and female reproductive tracts. So all of these are sort of the ciliary systems that are found in humans. The environments are pretty complex. You have these non-Newtonian fluid environments like the mucus and stuff. But what's remarkable is that if you wanted to study ciliary coordination, you could even look at simpler systems, for example, microorganisms. And what's remarkable is that nature has conserved the structure of cilia across these evolutionary timescales. So if you took the cross-section of a cilium in what's found in a human as compared to what's found in these organisms, they're pretty much the same. So on the leftmost frame, you see a bunch of sperm cells which are swimming, and you see that as they approach each other, they start to build their tails in phase. And on the second frame is
Anup, I don't know if this Anup.
A single-celled alga called the Chlamydomonas, which has this breaststroke kind of motion. It uses that to swim. So you see that this is a relatively dense carpet, and it has these. Now note that these are not neuronally connected cilia. Orchestrated. The behavior seems. So on the bottom right frame is an example of the kind of ciliary carpets, a dense one, which we hope to study. Right. So specifically, like I just said in the previous slide, what we want to study is the collective behavior which emerges in carpets of cilia. And this is at the scale of tissues, the length scale of tissues. So if you noticed in the previous video, all of these cells, which I showed, you saw that they constantly kept beating. There was no coasting like motion. Now, these are all immersed in basically water, right? This is not how humans swim. So there is some funny flow physics going on, and that's what I'm going to do a quick primer on right now, which is that we all know the Navier-Stokes equations. They govern the motion of all moving fluids. And they're basically Newton's second law, F equals ma applied to a fluid parcel. So there is a dimensionless group called the Reynolds number, which is probably the most important dimensionless number fluid mechanics study. And it's simply a ratio of the inertial to the viscous forces. It quantifies the relative importance. So for example, if I were to compute the Reynolds number of a human swimmer, like Michael Phelps, say, he swims at about 2 meters per second and he's about 2 meters long. It would be of the order of 10 to the 6. Planes and submarines and so on, they have numbers which are even higher. Now, when I compute the Reynolds number for a really tiny microorganism, which has a length of just in the order of microns, turns out the Reynolds number is very slow. So this is telling us that the viscous forces are much, much more important than inertial forces. But there is also a very classic interpretation of Reynolds number as some sort of a dimensionless coasting distance, which is, if you are a swimmer, as human swimmers, we can deal with all this. We can imagine this through experience, that if I'm doing my swimming motion and I just stop applying force on the fluid, then I'm going to coast forward purely because of my inertia, right? So it turns out that you could directly relate this coasting distance to the Reynolds number of the environment you're swimming in. And as you can see here, a human, we know from experience, we coast for about 1 body length, so the order of a meter. Whereas for a microorganism, that's like 10 to the negative 12 times. So basically a micron scale organism is going to move about 1 angstrom, which is like atomic length scale. So there is no inertia in these flows. And in fact, you could work this out mathematically. You could rescale your equations, write it in terms of the Reynolds number, and what you get is the Stokes equations. So what's special about Stokes equations is they're linear and they do not have a time term. There is no unsteady term. So because of this, a direct consequence of this is called kinematic reversibility, which means that if you're a single degree of freedom system, like a scallop, then when you apply force on the fluid inward, and let's say you get displaced in this direction, then when you're opening up your shell, basically you're reversing the sign of all these terms, which means that you're just going to go right back to where you came from, no matter how fast or slow you did it. So this means that organisms at small length scales have to use different swimming strategies. Winning 9 Olympic golds at low Reynolds number is a humongous task. So this is also seen in cilia, wherein a cilium has an asymmetric beat. Basically, it moves forward like a rigid rod and comes back like a flexible filament. More on this just in a bit. So before moving on to cilia, there are two classic solutions of the Stokes equations, which we will make use of today. One is the impulse response of the Stokes equations, which is that I have a point force. I have a point force here in the fluid domain, and the fluid domain is infinite. And the solution of this can be worked out using Fourier transform techniques. And it's basically the Green's function of the Stokes equations. And it can be worked out to be of this form. It depends only on the geometry, basically, where you're measuring the flow versus where the Stokes rate is. Now, in biology, cilia and flagella usually, they exist, they're attached to some sort of a surface. So what's more relevant to look at is what's known as a Blakelet, which is you have a force monopole in the domain, but instead of the domain being infinitely large, you have a bounding no-slip wall. So these are the sample flow fields induced by like a force monopole. As you can see for the Blakelet, which is the case in the end, the velocity at z equals 0, which is where the wall is, it satisfies the no-slip condition. And the way to derive this was done classically in some work by Blake, where you take the Stokes slit, which is the same term here, and then you add a bunch of higher order singularities and put them at the image location. This was inspired from work in electrostatics for reconstruct image systems. So now we have enough background to move on to talk about ciliary synchronization. Obviously, we're not the first ones to do it. And like I showed, cilia are generally immersed in fluid environment. So there is plenty of evidence in the literature which suggests that hydrodynamic coupling is actually a very important factor when it comes to synchronizing cilia. So the most biophysically complete models, they model each filament, each cilium as basically a semi-flexible filament, which has beating motion like is shown in this video. And Elgeti and Gomper in a classic study, they put a bunch of these cilia together and started them off fully synchronized, like you can see here. And what happens as you step through time is that you see a transient phase where the beat seems to be incoherent. And then finally, you see these metachronal wave-like patterns emerge. So if you were to look from the top down and map the tip displacement of each of these cilia, each of these filaments, you would see waves like this. So in this representation, the waves are clearer. Now, these are filament models are nice, but they also come with problems, which is that they're a high degree of freedom system. You would have to spend a lot of computational resources, and at some point, it just becomes intractable. So a second class of models, known as rotor models, have become very popular to model cilia, which basically use the idea that you have a filament, and you can just abstract that out as a bead moving in a circular trajectory. And these have nice properties, like it's easier to computationally scale, and it's also analytically tractable. So people have looked at this as well. In fact, in a recent work, Meng and co-authors and Soloviev and Friedrich, in fact, showed that a wave-like state imposed on a system like this is actually stable. But it's still not clear the bridge about how, if I were to start from a random initial condition, where do I go? Do I end up at the wave state? And that's the question we set out to answer. And we'll be using the rotor model. So to sort of reiterate the idea behind it, you have a cilium, which, like I mentioned, has an asymmetric stroke. It's a rigid rod on its way forward, and then it's just flexible on its way back. We can model that as basically a rigid bead moving in a perfectly circular trajectory. And to sort of capture this asymmetric bead pattern, what we can do is we can make the force magnitude phase dependent. And in general, it can be expressed as a sum in a Fourier series form like this, where you have M modes and you have amplitudes alpha M and beta M. So this is a sample force profile where I have F equals 1 plus half cosine plus half sine. Notice that the 1 plus is basically to ensure that it's always moving in the same direction. And this is a sample flow field induced. So now note here that locations where the force is high, it moves through very quickly, and the locations along the trajectory where the force magnitude is low, it spends a lot more time. So if you were to take the weighted sum of the flow, which is the net pumping over one cycle, it's actually just zero because of time reversal symmetry. Now, a fair question to ask when you look at this, in fact, is you're abstracting the bead, you're abstracting a solid filament with like just a point force. Isn't that the traditional spherical curve model that physicists get accused of using all the time? And the answer to that was actually found in experiments from the group of Ray Goldstein at Cambridge, where what they did is they took the Chlamydomonas cell, which has two flagella, like I showed earlier, it swims in the breaststroke fashion. They did micro PIV experiments and basically mapped out the flow field around it. And then what they did is they just approximated this entire organism using 3 Stokes slits, one for each flagellum, which are the green arrows, and one for the body itself, which is marked out in red. And as you can see, the nature of the flow is very, very well captured by just this simple like force monopole model. So this indicates that it's actually a good representation of a cilium as a single rotor. It does seem to capture all the important flow features. So now I'm going to set up the problem that we want to solve. So we want to solve for a bunch of cilia placed in a square lattice with lattice spacing d. And all of these guys are on the same layer at a height h above a no-slip boundary. And all of them are immersed in a viscous fluid. So that's how they communicate with each other. So if I take any individual cilium, let's say the jade cilium, it's characterized by its angular phase theta. So its position is simply the location of the center of the trajectory and the vector sum of that in the normal direction. And then the velocity of the bead, you can just take a first derivative of the position with respect to time and you get the velocity. Now on any cilium, there are 3 forces acting. One is the active force, which we prescribe to be strictly tangential. The second is the constraint force, which is to make sure that the bead stays on the circular trajectory and doesn't move about. And the third is a drag force. So the cilium is beating about in a fluid, so it's going to encounter some drag. And this drag is proportional to its relative velocity with the background fluid. So the first term in the drag expression is actually the velocity of the bead itself. The second term comes because of its interaction with all the other cilia in the domain. So that's the induced flow. Now we know all the forces, so we can just do a force balance using Newton's second law, which just says that the active force and the normal force have to be balanced out by the drag force. So we can take the inner products with the normal and tangential vectors, and taking specifically with respect to the tangential vector gives us the phase equation of motion. So phase equation of motion is saying something very simple and intuitive. It is that the phase velocity of any cilium, theta j dot, has 2 components. One is because of its intrinsic angular speed, which is imposed because of the tangential force that we prescribe. Note that this is phase dependent. This is a function of theta itself. And the second component is it arises due to the hydrodynamic interaction with all the other cilia in the domain. And we know how to compute the total background flow because remember, we are in Stokes environment. So it's linear, which means that the total flow induced at the location of the jade cilium is basically the sum of the contributions due to all other cilia in the domain. And the exact coupling is quantified by the Blake tensor, for which the expression is well known. Now, before moving on to the numerical simulations, I'd like to point out there are two periodic solutions to this Gowning equation. One is the fully synchronized state. So imagine you have all cilia locked in the same phase, and you let them go basically. So at any instant in time, the velocity induced is tangential, and this continues sort of for all time. So each cilium basically sees the same neighborhood around it, which means nothing really changes. So there is no reason for the system to fall out of the state. So this is one periodic solution. Now the second one is an isotropic state, which is a little more subtle. So what we do is we define a new phase using a nonlinear transformation. And this new phase satisfies this condition, which basically says that the intrinsic angular frequency of a cilium in this state is constant, omega 0, which in fact is the beating frequency of an individual isolated cilium. Right, so we define the isotropic state as one from which is drawn from a uniformly random distribution of phi. So what I just said in many words, it's basically captured in this picture here, which says that if you take a uniformly random distribution of phi, it's a flat line, you end up with some distribution of theta. And what's nice about this is that the region along the trajectory where most number of cilia are is actually the region where the force is the lowest, right? And most cilia occupy the location along the trajectory where the force is minimum. So this is something quite intuitive. So the question we now ask is, are these periodic solutions stable? Now, to answer that numerically, what one would ideally do is you have this equation to step through time. You need to evaluate the induced velocity here. To numerically sort of integrate this equation and see what happens in the dynamics, what one would want to do is solve for this induced velocity v at the location of the jth cilium, and then we would know everything that happens. So here I'm showing two schematics of the two states I showed in the previous slide, where it's just a system of 5x5 cilia. And here the flow fields are quite straightforward to compute because we know the Blake tensor. But the problem now is, if you notice, this Blake tensor couples each cilium in the domain with each other cilium in the domain. So for each cilium, you'd have to do the summation over all the other cilia in the domain, which means that when you have to integrate these number of equations of motion for each cilium, this is a classic quadratic scaling. So this happens because hydrodynamic interaction has a slow decay. So we quickly run into trouble when we try to simulate large carpets of cilia, which is that they become intractable. The time now grows quadratically, simulation time. So to do this, the first step was to come up with an algorithm which enables us to do large-scale simulations. So I'm going to talk about that next. So our algorithm uses two key ideas. One is that we truncate hydrodynamic interaction. So it is well known that in the presence of a wall, there is some amount of hydrodynamic screening, which means that a classic 1 over r kind of decay now decays as 1 over r squared, r being the distance between where you're evaluating the velocity and where your singularity is. So we are going to have a fundamental domain, which is going to consist of some number of cilia. And then we are going to impose periodicity in the x and y directions, which is shown by the gray boxes here. And then to truncate, what I'm going to do is I'm going to draw an interaction stencil around each cilium and see what happens to the induced velocity at the location of the cilium at the center, like I'm showing here in a zoom view, due to increasing number of shells. So one shell would enclose, like, including the focal cilium, it would have 9 cilia. 2 shells would have 25, and so on. So what I'm showing here now is the top row is basically the cosine field. So I have 151 by 151 cilia, so it's not possible to represent it by circles anymore. So I'm plotting the cosine of the face. Each pixel is the cosine of the face of the cilium at that location. I'm going to compute the induced velocity at some guy in the center as a function of increasing number of shells. So what we see is quite sensible, which is, and I do this for 3 different states. So what we see basically says that as you increase the number of shells, as you keep increasing the number of interacting neighbors, your flow speed induced at the focal cilium is going to converge to a true solution. And this true solution is something we get from the fast multipole method, which we got from Vanyan at Flatiron Institute. So as you can see, for all these 3 states, the flow speed at the focal cilium converges to the true value based on FMM. So this tells us that although there is no free lunch, we have to live with some error. It's fine to truncate. The second idea is to use lookup, which is that you would compute the Blake, you would evaluate the Blake tensor at each and every time step, but that's not really required because all of these guys are moving in perfectly circular periodic trajectories. So what we can do is actually pre-compute this effect for each pair, which includes the focal cilium, and basically pre-compute and record the induced velocity due to each other cilium on the focal cilium. To do this, we first discretize each trajectory into some n theta number of parts. And then we simply evaluate this once before running the algorithm and record this in a table. And at runtime, all we have to do now is do a 2D linear interpolation and get the induced velocity out. So with these 2 steps, we are now in a position to run simulations, and we are going to use 151 by 151 cilia, like I said earlier. They are arranged in a square lattice, and periodicity is imposed in both x and y directions. We are going to use Runge-Kutta 4th order method, and we are going to integrate for 100 arbitrary time units. So before I show you the results, I have to tell you how I quantify the emergence of these wave patterns. I do this using something called the Kuramoto order parameter, which is a very classic metric used in phase oscillator studies. All it says is that it has this form, and all it's saying is if you have a bunch of phase oscillators which are in the same phase, that is, they're phase locked, then this summation and its absolute value is just going to be n, which means that the Kuramoto order parameter, which is the magnitude of this guy, is just 1. And if there is phase disorder, each guy is pointing its own way, then this summation is going to be very small. In the limiting case, it's just going to be 0. And the Kuramoto order parameter is 0. So what's important to remember here is that a Kuramoto order parameter of 1 indicates full phase locking, and Kuramoto order parameter of 0 is complete disorder. Right, so I'm going to let these simulations play. We ran these simulations using 2 force harmonics that I showed you first, m equals 1 and 2, and we'll see what happens. So you see that starting from an isotropic state, the Kuramoto order parameter is 0, and then it goes through a transient phase, and then we see the emergence of these wave-like patterns in the domain. This is really nice. This is what we are hoping to look for, but the problem here is that the Kuramoto order parameter does not seem to contain any useful information at this point. So, and it actually quite makes sense because initially there is phase disorder, so it's 0, but even towards the end, the spatial organization has changed, but the distribution, if you look at it, in a wave and in an isotropic case aren't too different. You would have a distribution between 0 and 2 pi. So even this guy sort of just averages out to 0. So this is a problem, which is that this metric does not seem to work. But so we had to come up with a metric that actually does capture the emergence of the spatial coherence. So for that, what we do first is instead of computing the Kuramoto order parameter summed over all the cilia, we're going to compute something called the order field, which is the same as the Kuramoto order parameter, except for the fact that I'm just going to compute this number locally, which is at all of these cilia are arranged on a grid. So I'm going to just pick the closest neighbors of J, compute Y1, and then I can move over to this order field, so-called. So this second plot here is the order field, which just shows local synchrony, right? And then I can actually draw lines starting from the origin at different angles alpha, and I can compute the measure of spatial I can measure the average of the value of this order field along any given direction alpha, take its magnitude, and that gives me P alpha. So now I've set myself up for a good polar coordinate representation of this. So if I have phase disorder, which is the isotropic state, and I do this polar plot of alpha and P alpha, what I see is that all of these points are huddled around the center, close to magnitude 0, and they're roughly symmetric, which makes sense in the case of isotropic state. But what's amazing is that when you, so this is synthetically obtained wave state. So if I compute the same metric in this wave state, then you can clearly see that the points stretch out in the direction where the spatial correlation is maximum. So there is most phase coherence in the cilia along the direction pi over 4 in this case. And that's exactly what you see. So to quantify this, you could go one step higher and actually use PCA to fit an ellipse through it. And you can measure its eccentricity and inclination to the horizontal. So we are going to now use this metric to look at our numerical simulations. And this is really nice now, because initially in the isotropic state, it started out pretty much symmetric about the origin, and then you can see it's getting squished into an ellipse. And you can also track that in the Kuramoto eccentricity, which you see sort of begins to stabilize around this value here. And here you can clearly see the ellipse as the waves have emerged here. Now, this effect is even more pronounced in case of the second force harmonic. Oops. Where in fact the circle really stretches out into the ellipse very quick. And the Kuramoto eccentricity and angle also stabilize very quickly. Just a quick reminder that Kuramoto angle is basically the inclination of the first principal component with the horizontal. And so now this is nice. We know exactly how to measure spatial coherence, especially one which looks like waves. So we can do one more thing. We know that in biology, cilia generally coordinate to achieve net pumping. So what we do is, we know that the pumping due to a Blakelet or a Stokes-Slit of strength F at a height h above a wall is basically proportional to its strength. So now we can compute the pumping due to the entire carpet by simply summing the individual contributions. Again, this is Stokes' law, so we can do this. And then what I'm going to do as a final step is I'm basically going to position myself somewhere in the fluid domain or in the ciliary carpet and wait and watch how much of fluid washes over me, which is basically the cumulative pumping. So I can do one more step, which is to measure the pumping in the direction of the wave propagation, which I got from my Kuramoto ellipse. And what we see is that there's a pronounced pumping once the wave state emerges. So I'm showing here the pumping with respect to time. And as you can see at 100 time units, which is how long the simulation was, it starts to pump fluid. Now, we also noticed that the force harmonic, which gave waves of thicker wavelength, like I showed on the previous slide, seems to be poorer at pumping fluid compared to the first force harmonic. And that will be it about the numerical simulation part of this. Now, the second goal was to study infinitely large ciliary carpets, because even this simulation, although it's large, it still has limitations in the sense that we can't have 1,000 by 1,000 cilia. So it made sense to develop a continuum theory, and we did this in collaboration with an awesome physicist from Flatiron Institute, Sebastian. More about him in a bit. So first, I want to convey the idea of a continuum of cilia. So what we want to do is we basically zoom out infinitely, and now there are infinitely many cilia in the domain. And at this point, you can't really differentiate individual cilia. You just have a layer which is generating some stress due to the activity of each cilia, right? So we can write down the Stokes equations above and below this layer. I'm going to denote those regions with plus and minus. We can solve Stokes equations independently in each of these. And then what we can do is we can work in the existence of the ciliary carpet using boundary conditions. So the first, I'm going to go through these boundary conditions one by one. The first is that we want pressure to remain finite in the far field. It can't blow up to infinity. So as z tends to infinity, pressure has to converge. The second condition is that of no slip at the wall, which is standard. Now, this is where the existence of the ciliary layer comes in, which is that we say there is no jump in the velocity between the bottom and the top layer. So in the limit of z tends to h plus and h minus, the flow has to be smooth. There is no jump. And the final one is to match retraction, which is that you have this layer of fluid which is driven by these active, or which is set into motion by the activity of the cilia. So we're going to say that the stress jump is actually the force exerted by the cilia in the domain. So what's nice about this formulation is that we can now move to the Fourier space, where notice that the unique thing about this Fourier transform is it's two-dimensional. I have a 3D fluid velocity u, which I'm going to Fourier transform only in the x and y directions. I'm using the wave vector k, which is just kx and ky. Now writing these, I can write down the Stokes equations in Fourier space, which are really nice constant coefficient ODEs, which we learn in the first graduate class in math how to solve. So we can actually solve for this, and we get a bunch of these arbitrary constants that depend on the wave number k though. And then we can use the boundary conditions to determine all of these A, B, C, D, and E, F. And we know exactly what the in-plane velocity V is. Sorry, I forgot to mention that. V hat is the Fourier transform of the in-plane velocity. And this K hat is basically the Stokes kernel. So now that we have an analytical expression for the velocity induced in the ciliary layer, we can actually do nice things like do linear stability analysis of the isotropic state. So what we had earlier was a discrete description of the cilia, which is you have these individual cilia on a carpet, and we were tracking their phase, theta or equivalently phi. So now we introduce these macroscopic, this macroscopic quantity, which actually is very similar to the Kuramoto order field, which I described earlier, only that we now also allow for higher moments of it. So we have yn, which is the summation of e to the in phi j. For a finite system, we could scale this by n as well. And what I really want to convey is that this macroscopic variable is basically tracking the different kinds of order that could exist in a system. So instead of looking at the face of each cilium, now we are going to ask questions about the collective, which is, like I said earlier, in n equals 1 case, this is just the Kuramoto order parameter, which is 1 when all the cilia are in phase. Similarly, n equals 2 captures antiphase behavior, and it's classically known as the nematic order parameter. So it is 1 in case of antiphase sort of synchrony, and so on. You could track many different kinds of order. So this also lets us define what kinds of order exist in the collection of cilia. So we can now move on to a continuum description, where the microscopic variable, which was the angular phase, has been replaced by this macroscopic variable yn, which are orders of the moment field. So a quick recipe for how to do this linearization is here. The derivation is quite tedious, so if anyone has questions, please interrupt me. I'm happy to go over the derivation. But the basic recipe is as follows. You define the moment fields as follows. Then you can actually take the first derivative of this and write down its time evolution. Now in the isotropic state, we define the isotropic steady state as one in which yn is 0 for all moments except the 0th moment, because in 0th moment, you're just basically summing a bunch of ones. So we normalize that to the ciliary density in the domain. And then the classic recipe for linearizing equations is we perturb about the base state, and we know this definition of the base state. Then what's left to evaluate is again the velocity, but now we know it from the Fourier space. So we can introduce a perturbation in the continuous force density, which has a similar formulation to the moment field. And then we can find the perturbations in it and actually just relate it to the induced velocity v, because we know that v hat equals k hat times f hat. The only difference here is that now we are working in the Fourier space, but that's fine. So finally, we end up with an equation for the time evolution of the perturbation, which is a matrix equation with the coupling matrix psi. And what we're going to do is that get the leading eigenvalue, that is the eigenvalue with the largest real part. And that is going to give us the growth rate of the perturbation in Fourier space. So we can do that. Notice that this theory is linear in the sense that for any moment field, oops, sorry, there's a typo here. This should be delta y hat of k. It's not an x anymore. So each kth wave number, each wave number pair k does not interact with all the other wave numbers. It's linear in that sense. So we can go ahead and actually compute the growth rates for all different modes. So this plot is, each pixel here is the growth rate obtained by solving one of these equations at wave number pair kx, ky. I can do this for many number of wave number pairs and get this growth rate plot out. And I can repeat this exercise for different force harmonics. So notice that even in the numerical simulations, I was showing just the first and second force harmonics because we found that the higher force harmonics don't lead to wave-like states. So the linear stability analysis is telling us that the isotropic state is linearly unstable, and to at least a qualitative level, this matches what the numerical simulations say, but we were greedy, so we wanted more. So what you could do now is that you could start with a moment field, which is also of the size 151 by 151, say a discrete version of the moment field. And what you could do is go to the Fourier space, grow the perturbation in the Fourier space, because we now exactly know what those numbers look like, and actually see how this system would evolve in time. So I guess the poster frame sort of spoiled it, but what we see is that for the first and second force harmonics again, we can see that this Kuramoto ellipse sort of stretches out, and the Kuramoto eccentricity also stabilizes. And we see some spatial coherence, which looks kind of similar to what we saw from the numerical simulations. Now, we can also do this for the second force harmonic, and again, notice that we see waves which are roughly of the same orientation that the numerical simulations told us. So with this formulation, we can actually compare the numerical simulations and the theory, not just qualitatively, but even quantitatively. So what I'm showing in the first column of the slide is the growth rates, and on it, these little black pepper dots are basically the Fourier transform obtained from the numerical simulations. I'm just overlaying them. I take some cutoff, and basically I'm showing which ones are most unstable. And the theory and the numerics agree here. But this is still sort of qualitative. And then in the second column, I'm showing the Kuramoto order fields obtained from theory, which is the first half of the frame, and from numerics. And as you can see, the structures here that you see are very similar between these two, like much better than we were actually hoping to see, because this is just a linear theory. So this is quite amazing. And then we can actually go ahead and compute the Kuramoto ellipse. So again, here the black dots are obtained from numerical simulations. And then what I do is I take multiple realizations of this random order field, grow them, repeat this exercise across 200 different initial conditions, and it's pretty cheap to do. And then I get this gray shaded Kuramoto ellipse. The dark gray is basically the mean of all these. And then I can actually plot out the range and the mean of this guy, and overlay again the black dots coming from numerical simulations. And this is really nice. This shows us that two approaches which look very different. In the numerical simulations, we were directly integrating the phase equations, whereas here we linearize, we compute the growth rate, and we're tracking the orders of moment fields. And both of them seem to give something matching quite remarkably well. So we can do this exercise for the second periodic solution as well, which is the fully synchronized state. Again, I'm going to quickly lay down the recipe of this, which is that We don't need to introduce the moment order fields here because we can directly perturb the phase itself. The fully synchronized state is nice in that sense. So I can get rid of the index because all rotors basically look the same in the fully synchronized state. And then I can actually compute the velocity. And we even know through intuitive arguments that the velocity induced at any cilium is basically tangential to its trajectory. So plugging this into the Gaussian equation basically tells me that at the fully synchronized state, the phase velocity of any cilium is basically just amplified. It's multiplied by a constant times the imposed or driving frequency of the cilium itself. And then I can introduce a perturbation, and I can follow the same recipe that I did for the isotropic state, which is to define a continuous force density, find perturbations, introduce perturbation in this, and find the resulting perturbation and velocity in the Fourier space. And then I can again compute the cycle average growth rate. Now, notice that the isotropic state did not have any well-defined periodic cycle by itself, but the fully synchronized state does as a result of this equation. So the T here at the bottom is the time period resulting from this phase velocity. And this is classically known as the Floquet multiplier. It's the cycle averaged growth rate of the perturbation. Right? And I can do this again. This is for each wave number pair. I can compute this. And I can repeat this for all the force harmonics. And again, plot it with kx and ky. And we see that the first and second are again the most strongly unstable ones. So the fully synchronized state is also linearly unstable. Although notice that unlike the isotropic state, there are some modes which are actually stable. Now, again, we could compare this with the numerical simulations as well. And running the simulations here was much easier because we only need to run it for approximately one cycle. And we can compute the Floquet multiplier directly by taking a Fourier transform of the phase itself. We can do this for all wave numbers. And this is what the comparison looks like, where I'm doing it for the two force harmonics again. And this is what the theory predicts for the first force harmonic, and it's in pretty good agreement with what the numerics say. The last column of plots basically shows cross sections of these guys. The solid line is from the theory and the dotted line is from the numerics. And we see that it matches quite well, except for the fact that continuum theory, by its very definition, it coarse grains over the smaller length scale, which means we lose some information at higher wave numbers. So the math seems to be much better here. And that basically brings me to the end of this talk. I'll end with with a summary and contributions of our work with some future outlook. So what we did was we introduced an algorithm that enables large-scale simulations using a minimal model. And then we observed emergence of metacanal coordination, and we also found a metric to actually quantify this. What we found was that the force harmonic, like I showed, was the most important factor when it came to the nature of synchrony that emerged. First and second force harmonics led to synchrony, but none of the others that we checked did. And this actually matches qualitatively well with experimental measurements of the force profile of a cilium. And then we introduced the Kuramoto ellipse as a useful metric for quantifying spatial coherence. And finally, we developed a continuum theory, which allowed us to probe the stability of the isotropic and fully synchronized states in the limit of having infinitely many cilia. And we were able to match the results from the linear theory and fully nonlinear simulations, not just qualitatively, but also quantitatively. That's basically the end of my talk, and I'd be happy to take questions.
Thank you so much, Anup.